Welcome back to the deep dive. Today, we are shifting gears a little bit. We spend so much of our time in our day-to-day lives thinking about speed.
Absolutely.
How fast can I get this done? How quickly can I get energy? And in previous deep dives, we've really looked at metabolism through that exact lens glycolysis, the sprint.
100-meter dash.
Exactly. But today, we're looking at the other side of the coin. We're looking at the metabolic marathon.
It's the perfect distinction, really. If glucose is the cash in your wallet that you spend at the corner store.
Right, quick and easy.
Then the topic we're covering today is the gold bullion in the vault. It's dense, it's hard to move, but it is where the real wealth is stored. We are talking about fatty acid catabolism.
The burning of fat. And this feels particularly relevant because physiologically speaking, most of us are carrying around enough of this fuel to survive for weeks.
Weeks, maybe even months, without a single bite of food.
But accessing it isn't as simple as just deciding to burn fat. There's a whole process.
Not at all. It is a system built for endurance, not for, urgency. When you really look at the biochemistry, as we will today, you see a system that is incredibly deliberate.
Okay.
It's highly regulated. It guards against waste. It's a completely different philosophy from the explosive use-it-now energy of sugar burning.
And to guide us through this, we are synthesizing two absolute titans of the field. We have Leninger's Principles of Biochemistry, specifically the chapter on fatty acid catabolism, which gives us that beautiful mechanistic chapter on fatty acid oxidation and ketogenesis, which grounds us in the medical reality of what happens in the hospital when this elegant system breaks.
And, as always, a reminder to you, the listener, we're building a conceptual map here. We want to help you see the logic of the system.
Right.
So that when you go back to the textbooks, and you absolutely should go back to textbooks, you're not just memorizing enzymes, you're understanding the story.
Let's start with the mission then. The goal for this deep dive is to track a single fatty acid molecule from its dormant state, locked away in a fat cell.
In the vault.
In the vault, all the way to the mitochondrial furnace where it gets chopped up into energy. And it sounds simple, but there are walls and guards and transport systems every single step of the way.
It is a massive logistical challenge. Just think about it for a second. You have a fuel, fat, that doesn't dissolve in the blood, which is mostly water.
Right, oil and water.
You have to move it to a different organ. You have to get it into a cell. Then you have to get it across an organelle membrane that's specifically designed to be impermeable to it. And only then can you start to burn it.
Okay, so let's begin at the source. The wake-up call.
Yeah.
We are in a adipocyte, a fat cell. Set the scene for us. What does this cell look like when we're well-fed and happy?
If you look at an adipocyte under a microscope, it's pretty distinct from almost any other cell in the body. It looks like a massive signet ring.
A signet ring.
Yeah. The nucleus and all the other organelles, the mitochondria, the ER, they're all squashed to the very edge. And the center is just dominated by one massive lipid droplet.
A giant glob of grease.
Essentially, yes. It's filled with triacylglycerols, or tags. That's the storage form of fat. But I think the common misconception is that this droplet is just a passive blob of oil, like a bubble in a lava lamp.
It's not.
It's not. It is a fortress.
A fortress? How so?
It is heavily, heavily guarded. The entire surface of this lipid droplet is coated in a layer of proteins. They're called perlipins.
Perlipins. Sounds like peril.
Well, it prevents peril, in a way.
Yeah.
These proteins, they stand shoulder to shoulder, completely locking the surface. They physically block the cellular enzymes, the lipases, from getting to the fat.
So in a fed state, the warehouse is on lockdown.
Complete lockdown. You cannot withdraw the gold. It's just not accessible.
Okay, so the guards are standing firm. But now the environment changes. Let's say I haven't eaten in 12 hours. Or, to use a more dramatic example, a lion walks into the room.
Right. Fight or flight.
I need energy, and I need it now. What is the signal that opens the vault?
You need a hormonal trigger. If you're fasting, that trigger is glucagon. If it's the lion, it's epinephrine or adrenaline.
And these hormones bind to receptors on the outside of the fat cell, right?
Exactly. And this initiates a classic signal transduction cascade. We've seen this pattern a lot. The hormone doesn't actually enter the cell.
It just rings the doorbell.
That's a perfect way to put it. It rings the doorbell, which activates an enzyme called adenylyl cyclase. That enzyme then churns out our second messenger, cyclic AMP or CAMP.
And CAMP wakes up our favorite cellular foreman, Protein kinase A, PKA.
Exactly. And PKA's job is to run around and phosphorylate things, essentially slapping a phosphate group onto specific proteins to change their shape or their function.
It's the activation switch.
It is. Now, usually textbooks will just say PKA activates the lipases, but Leninger gives us the specific, intricate choreography of the moment, and it involves a character that often gets left out of the story.
Okay, who's this mystery character?
It's a protein called CGI58.
CGI-58. Sounds like a droid from Star Wars.
It kind of acts like a key. In the resting state, when the cell is just storing fat, CGI58 is trapped. It is physically bound to the paralipin protein on the surface of the droplet.
So the guard is holding the key?
The guard is holding the key, and as long as it's stuck there, fat breakdown cannot start.
Okay, so PKA arrives on the scene.
PKA comes in, and it does two crucial things. First, it phosphorylates the paralipin, the guard itself. This causes a massive conformational change. The paralipin twists, and in doing so, it releases CGI58.
The key is dropped.
And CGI58 is immediately recruited to the first enzyme in the chain, ATGL, which stands for adipose triacylglycerol lipase.
Right.
Without CGI58, ATGL is totally dormant, but with it, it becomes a molecular chainsaw.
I love that detail. It's not just turning it on. It's a specific release mechanism. So ATGL starts the work. But we're breaking down a triacylglycerol. That means 3 fatty acids attached to a glycerol backbone.
And just like you can't take all the legs off a stool at once, you have to cut them one by one. And this is regulated by 3 specific enzymes. First, as we said, ATGL, activated by CGI58, cuts the first fatty acid.
So we go from a triacylglycerol to a disoglycerol. 2 fatty acids left.
Correct. Now, the second thing PKA did was phosphorylate another enzyme, one called hormone-sensitive lipase, or HSL.
Okay, so this one responds directly to the hormone signal.
That's right. And HSL, now activated, swoops in and cuts the second fatty acid.
Now we have a monoacylglycerol, just one left.
And finally, a third enzyme, which is just constitutively active, called MGL, for monoacylglycerol lipase, cuts the last one.
So it's a dismantling crew. ATGL, then HSL, then MGL. 3 different enzymes. Why can't one enzyme just go chop, chop, chop?
It's all about specificity and control. Each enzyme is structurally tuned to the specific shape of its substrate. A generic lipase would be messy, less efficient, and much harder to regulate.
I see.
By having 3, the cell has multiple control points. For example, HSL is the primary rate-limiting step for that second cut. It allows the cell to fine-tune exactly how much fat is being released at any given moment.
So the result of this whole demolition is 3 free fatty acids, FFAs, and 1 molecule of glycerol. The glycerol is the easy part, right?
Yeah. The glycerol is water-soluble. It just slips out of the cell, dissolves right into the blood, and floats over to the liver.
And what does the liver do with it?
The liver has a special enzyme, glycerol kinase, that can phosphorylate it. It can then use it to make a little bit of glucose through gluconeogenesis, or just burn it in glycolysis.
So glycerol is like a little bonbon of sugar potential.
Exactly. But it's a tiny, tiny fraction of the energy. The real energy is locked in those fatty acids. And here we hit our first major logistical problem.
Right.
We have released these long, oily, hydrophobic chains into the cytosol. They need to get to the muscles, to the heart, but the blood is, well, it's water.
And oil and water do not mix. If you just dumped fatty acids into the bloodstream, they'd clump together, they'd coalesce into droplets and block your capillaries. You'd have a fat embolism.
Precisely. The system would fail immediately. So we need a transport system. We need a taxi.
Enter serum albumin.
The great unsung hero of the circulatory system. Serum albumin is this massive protein produced by the liver. It actually makes up about half the protein in your blood serum.
Wow.
And it floats around acting as a molecular sponge for all sorts of hydrophobic things, but especially fatty acids.
So how does it carry the fat? Does it have like a little trunk?
It has specific hydrophobic pockets, these deep clefts in its surface, where the fatty acid tails can tuck themselves in, shielding them from the water.
So it's like an escort.
It is. A single molecule of albumin can carry up to 7 fatty acids at once.
That's a minivan, not a taxi.
It's absolutely a minivan. And it ferries these fatty acids through the blood, dropping them off at tissues that have high energy demands. Skeletal muscle, the heart, the renal cortex.
But there is one very important place that the minivan cannot go, or rather, one place the passengers can't get off, and that's the brain.
This is such a crucial point. Fatty acids cannot cross the blood-brain barrier. For one, they are bound to albumin, which is way too large to cross.
Okay.
And even if they weren't, The specific transport proteins just aren't there on the brain endothelium. So while your heart is happily chugging along on fat during a fast,
Your brain is screaming for
it's
glucose.
screaming for glucose. And this single constraint drives the entire logic of starvation metabolism, which we'll get to later with ketones.
Okay, let's stick with the muscle for now. The minivan pulls up to a muscle cell, a myocyte. The fatty acid hops off the albumin and enters the cell. We are now in the cytosol of the muscle. Can we just throw it into the fire?
Not yet. As Leninger really emphasizes, a free fatty acid is chemically, well, it's inert. It's a long hydrocarbon tail. It's very stable. It doesn't want to react.
It's happy as it is?
Exactly. To get it to enter metabolism, we have to activate it. We have to attach a kind of chemical handle that makes it reactive.
And that handle, I'm guessing, is coenzyme A.
That handle is coenzyme A. We're going to convert a fatty acid into a fatty acyl-CoA. The enzyme that does this is acyl-CoA synthetase. But I want to pause on the thermodynamics here because this is where the metabolic accounting gets really interesting.
It's the cost of doing business.
Exactly. To attach that CoA, we need energy. So we use ATP, but usually when enzymes use ATP, they hydrolyze it to ADP and a single phosphate. That releases about 30 kilojoules of energy.
That's the standard currency. It's like spending one ATP dollar.
It is. But attaching CoA to a fatty acid creates a thioester bond, and that is a very high energy bond. Spending just one ATP dollar by going to ADP isn't quite enough to make the reaction strictly irreversible.
So if we just went to ADP, the CoA might just fall off again. The reaction could go backwards.
Precisely. The equilibrium would be too close to the middle, so the cell does something much more drastic. The enzyme cleaves ATP not at the gamma phosphate, but at the alpha-beta bond. It breaks ATP down to AMP adenosine monophosphate and a molecule called pyrophosphate, or PPI.
That's a deep cut. That's like ripping the molecule in half.
It is. But here is the brilliant trick. That pyrophosphate, the PPI, is immediately attacked by another enzyme, inorganic pyrophosphatase, which shatters it into two separate phosphate molecules.
And why does that matter?
Because that second shattering releases a massive amount of energy. But more importantly, it removes one of the products, the PPI, from the equation. By destroying the byproduct, it pulls the initial reaction forward with such force that it effectively becomes irreversible.
It burns the bridge. There's no going back.
You cannot go back. But from a bookkeeping perspective, to get that AMP back up to ATP, you have to add two phosphates using two high-energy bonds. So the metabolic cost of activating one fatty acid is energetically equivalent to spending two ATP molecules.
It's a sunk cost. You have to pay $2 up front just to get into the club.
And this is important for our final tally later on. We are -2 ATP before we've even started burning anything.
Okay, so we've paid the cover charge. We have our activated fatty acyl-CoA sitting in the cytosol. But the furnace, the enzymes for beta oxidation, are inside the mitochondrial matrix. And standing in the way is the inner mitochondrial membrane.
Which is notoriously impermeable, especially to something like acyl-CoA. It's a wall, and this is a critical design feature. You don't want fat randomly drifting into the mitochondria and burning uncontrolled. You need a gatekeeper.
And the gatekeeper is the carnitine shuttle.
I like to think of this as the nightclub bouncer system. It's a great analogy.
Okay, walk us through it, the bouncer system.
All right. On the outer face of the mitochondrial membrane, you have the first bouncer. The enzyme is CPTI, which stands for carnitine palmitoyl transferase I.
CPTI, got it.
CPTI looks at the incoming acyl-CoA, and it basically says, you can't come in here with that CoA jacket on.
Wrong dress code?
Wrong dress code.
Yeah.
So it removes the CoA and replaces it with a different molecule called carnitine.
It gives it a VIP wristband.
It gives it a VIP wristband. Now, the molecule is acylcarnitine, and this specific molecule can pass through the membrane via a special translocase protein. It slides through the velvet rope into the mitochondrial matrix.
Okay, so we're inside the club now. But the enzymes inside, they don't recognize carnitine, do they?
Exactly. They're looking for that CoA jacket. So on the interface of the membrane, you have the second bouncer, CPT2.
CPT2.
CPT2 stops the acylcarnitine, takes the wristband off, removes the carnitine, and puts a fresh CoA jacket on from the mitochondrial pool of CoA.
So we are back to acyl-CoA, but now we're on the inside.
Yes. And the free carnitine that was removed is shuttled right back out to the cytosol to pick up the next guest.
This seems incredibly, almost needlessly complicated.
Right.
Why swap the CoA, move it, and then swap it back? Why not just have a transporter for acyl-CoA itself?
This is the absolute brilliance of the system. This is where the regulation happens. By requiring this swap, the cell creates a master control point. This shuttle is the rate-limiting step of fatty acid oxidation. This is where the decision is made, to burn or not to burn.
And who or what makes that decision?
A single powerful molecule called malonyl-CoA.
Now, wait a second. Malonyl-CoA, that's the starting material for making fat, right? If I eat a giant bowl of pasta, my insulin spikes, my body starts building fat, and the very first thing it makes is malonyl-CoA.
Exactly. And now think about the logic. If your cell is actively synthesizing fat because you have plenty of energy from that pasta,
Right.
the last thing you want to do is burn the fat at the same time. That would be a completely futile cycle. You'd be building a house while simultaneously setting it on fire.
It's just wasting energy. So how does the cell stop it?
Malonyl-CoA is a potent allosteric inhibitor of CPTI, that first bouncer at the door.
Ah, that is so elegant.
Yeah.
The mere presence of the building block for fat synthesis, malonyl-CoA, physically jams the door to the furnace.
It locks the gate. So when you're well-fed, malonyl-CoA is high, CPTI is blocked, and any fat that's activated stays in the cytosol to be stored or used for membrane synthesis.
And when you're starving?
When you're starving, insulin is low, fat synthesis stops, malonyl-CoA levels plummet, the inhibition on CPTI is lifted, and fat just floods into the mitochondria to be burned for energy.
It's a perfect binary switch controlled by the intermediate of the opposing pathway.
It is cellular logic at its absolute finest, compartmentalization and reciprocal regulation.
Okay, so let's assume the gain is open, we are starving, malonyl-CoA is gone. Our fatty acid, let's use palmitate, a standard 16-carbon chain, as our example, is now inside the mitochondrial matrix. It's ready to enter the grinder. Beta oxidation.
The metabolic grinder. This is the heart of the deep dive, and the best way to visualize this is slicing a loaf of bread or maybe a salami. You have this long chain of carbons.
And we want to cut it into 2 carbon pieces.
Exactly, 2 carbon pieces of acetyl-CoA.
Why 2 carbons?
Because acetyl-CoA is the universal fuel for the Krebs cycle, the citric acid cycle. It's the common currency that plugs into the sexual furnace.
So we slice it 2 carbons at a time, and it's called beta oxidation because the chemistry happens at the beta carbon,
which is the C3 position, counting from the CoA end. We are going to attack and weaken the bond between the alpha carbon C2 and the beta carbon C3 in order to snap it.
And Leninger describes this as a 4-step spiral, a repeating process.
A beautiful repeating loop. It's always the same 4 steps. Oxidize, hydrate, oxidize, cleave. But let's break down the why of these steps because the chemistry really explains the logic.
Okay.
We start with a carbon-carbon single bond between alpha and beta. It's a rock. It's stable. You can't just hit it with a hammer and break it. You have to chemically destabilize it first.
So step 1.
Dehydrogenation, which is a form of oxidation. The enzyme acyl-CoA dehydrogenase comes in. It removes 2 protons and 2 electrons from the chain, one from the alpha and one from the beta carbon.
It steals electrons.
It does, and it gives them to an electron carrier, FAD, making FADH2. This forces the alpha and beta carbons to form a double bond, a transalpine, to satisfy their valency.
So we've turned a stable single bond into a more reactive double bond. Why? What does that get us?
Because you can add water across a double bond. You can't just add water to a simple single bond.
Yeah.
And that sets up step 2.
Step 2, hydration.
Right. The enzyme enoyl-CoA hydratase adds a molecule of water, H2O, across that new double bond. This places a hydroxyl group, an 8OH group, onto the beta carbon.
So now we have an alcohol at the beta position.
Exactly. But an alcohol bond is still pretty strong. We need to make it even more fragile. So that brings us to step 3, another dehydrogenation.
We oxidize it again.
We oxidize it again. The enzyme beta-hydroxycyl-CoA dehydrogenase comes in and steals 2 more electrons and a proton from that hydroxyl group.
And gives them to NAD plus 8O, making NADH this time.
Correct. And what happens to the alcohol when you oxidize it?
It becomes a ketone.
Yeah.
A carbon double bonded to an oxygen.
Precisely. We now have a keto group at the beta position. So now look at the structure you've created. You have a carbonyl group. That's a C double bond O on the beta carbon. And you already had one on the alpha carbon from the original CoA end. This arrangement of 2 carbonyls separated by 1 carbon pulls electrons away from that central bond between alpha and beta. It creates immense electronic strain. The bond is now incredibly weak.
It's ready to snap.
It is primed to break. And that brings us to step 4, thiolysis. The enzyme sialase brings in a free coenzyme, a molecule from the matrix. The sulfur atom on that CoA acts as a nucleophile. It attacks that weakened bond and CR-seq.
And just falls off.
The 2 carbon end falls off as one molecule of acetyl-CoA, and you are left with a fatty acyl-CoA that is now exactly 2 carbons shorter than what you started with.
And that shorter chain just goes right back to step 1 and does it all over again.
And again and again. Oxidize, hydrate, oxidize, cleave. It's a spiral that repeats until the entire chain is gone.
Let's do the bank balance. Let's talk about the energy yield.
Yeah.
Because this is the ultra-marathon part of the story. How much energy do we actually get from one molecule of fat?
Okay, let's stick with our example, palmitate, which has 16 carbons. To chop a 16-carbon chain into 2 carbon blocks, how many cuts do you need to make?
Well, 16 divided by 2 is 8, so you get 8 pieces.
Yeah.
But if I have a loaf of bread and I want 8 slices, I only need to cut it 7 times.
Yeah.
The 7th and final cut gives me 2 pieces.
Exactly right. So palmitate goes through the beta oxidation spiral 7 times.
Okay, 7 turns.
Each turn of the spiral produces 1 FADH2 and 1 NADH. So from the spiral alone, we get 7 FADH2 and 7 NADH.
And those go directly to the electron transport chain to make ATP.
Correct. Plus, we have the 8 acetyl-CoA molecules that we produced. Each of those goes into the citric acid cycle. And each turn of that cycle generates, what, 3 NADH, 1 FADH2, and 1 ATP or GTP. It's a massive payout.
So you get the payout from the slicing and then a much bigger payout from burning the slices.
Exactly. And when you run all the math using the modern PO ratios, so about 2.5 ATP per NADH and 1.5 per FADH2, the grand total is a staggering 108 ATP.
108. From a single molecule of fat.
Well, wait, we have to subtract the upfront costs.
Right, the 2 ATP we spent on activation way back in the cytosol.
So the net yield is 106 ATP.
106. Now compare that to glucose. One molecule of glucose gives you what, about 30 or 32 ATP, depending on the shuttle used?
Correct. So fat is more than 3 times as energy dense per carbon. It's an unbelievably efficient way to store energy.
That completely explains why we store energy as fat. If we tried to store the same number of calories as glycogen, we'd be enormous.
And there's another critical factor, water. Glycogen is hydrophilic. For every gram of glycogen, you store about 3 to 4 grams of water with it. It's heavy and wet.
Okay.
Fat is anhydrous. It's hydrophobic. It packs tight with no extra water weight. It is the ultimate lightweight backpacking food for the body. And speaking of water, there's a fascinating byproduct mentioned in Linenger Box 17-1.
The grizzly bear.
Yes, the grizzly bear. When a bear hibernates, it doesn't eat or drink for months on end. How does it not die of dehydration?
I always just assumed it, recycled its water or its metabolism just slowed down so much it didn't need any.
Well, every living cell needs water to function. But look back at the chemistry we just described. When all those FADH2 and NADH molecules drop their electrons into the respiratory chain, what is the final electron acceptor at the very end?
Oxygen.
And what does oxygen become when it accepts electrons and protons?
H2O.
Water, metabolic water. The process of burning fat actually creates water as a final product. For the hibernating bear, the oxidation of its massive fat stores provides enough water to keep it hydrated all winter long.
That is mind-blowing. It's a self-contained life support system. It manufactures its own water supply from dry fat.
It is nature's efficiency at its absolute peak. However, we do have to address the exceptions to the rule. So far, we've been talking about nice, neat, saturated fats, straight chains. But in our diet, we eat olive oil, we eat fish, we eat unsaturated fats with double bonds.
The kinks in the chain.
And those kinks, they break the grinder. Remember step 1 of the spiral? Acetyl-CoA dehydrogenase specifically creates a trans double bond between the alpha and beta carbons. But the double bonds found in natural fats are usually in the cis configuration.
The shape is wrong. So the next enzyme, enoyl-CoA hydratase from step 2, it looks at that cis bond and says, I can't work with this. It doesn't fit in my active site.
Exactly. The process stalls, so the body needs a fix. It has an auxiliary enzyme called an isomerase. Its only job is to find that cis bond and physically flip it to the trans configuration and sometimes shift its position.
It basically straightens the kink just enough so the main machine can accept it and continue.
Right. And for polyunsaturated fats, which have multiple double bonds, it's even more complicated. You need another enzyme, a reductase, which uses NADPH to get rid of some of the extra double bonds. The takeaway here is the body has a toolkit of pliers and wrenches to handle the messy stuff and force it into the standard pathway.
There is one other messy fat that leads to a really cool piece of biochemical trivia, and that's odd-chain fatty acids.
Yes, most biological fats in animals are even numbered. But plants and some marine organisms can make odd-numbered chains like C15 or C17.
So what happens when you eat those? You chop 2, then 2, then 2, but eventually you're left with a 5-carbon piece. You make one last cut, and you're left with a 3-carbon piece at the end.
Propionyl-CoA. You can't cut 3 carbons into a 2-carbon piece. The grinder just stops.
So what do we do with this leftover 3-carbon tail? Just throw it away?
No, the body's not wasteful. We turn it into something useful. We convert it into succinyl-CoA, which is an intermediate of the citric acid cycle.
Okay, so it could just jump into the Krebs cycle.
It can, but the chemistry to do this is wild. It's a 3-step process. First, we add a carbon to it, a carboxylation reaction that uses the vitamin biotin as a cofactor.
Yummy.
Then we have to do this incredible rearrangement of the carbon skeleton. And this requires vitamin B12.
Ah, cobalamin.
Yes. This is one of only 2 reactions in the entire human body that requires coenzyme B12. It facilitates a very complex free radical rearrangement. It literally breaks the carbon skeleton apart and puts it back together in a new shape to form succinyl-CoA.
That sounds chemically very difficult and dangerous.
It is. But the result is this useful molecule, succinyl-CoA. And here is the Harper's insight, the key clinical pearl. Because succinyl-CoA enters the citric acid cycle, it can eventually be converted into oxaloacetate. And what can the liver do with oxaloacetate?
It can use it to make glucose in gluconeogenesis.
Right. Now, wait. We are taught in first-year biology, fat cannot be turned into sugar.
Right, because the conversion of pyruvate to acetyl-CoA is irreversible.
And that is 99.9% true. The acetyl-CoA that makes up the vast majority of the fat cannot become net glucose, but this tiny 3-carbon propionyl tail from odd-chain fatty acids is the one exception. It is the only part of a fatty acid that is glucogenic.
That is a fantastic gotcha question for a board exam. Okay, speaking of turning fat into other things, let's talk about the alternate economy. Section 5, ketogenesis.
Ketogenesis is so often misunderstood. People hear ketones and they immediately think of either fad diets or diabetic emergencies. But fundamentally, it is a brilliant survival adaptation for the brain.
And why does the brain need this special adaptation?
Because, as we established, the brain cannot use fatty acids for fuel. They don't cross the blood-brain barrier. So, in a normal state, the brain runs almost exclusively on glucose. But what happens if you are starving? I mean, truly starving. For days on end. Your glycogen stores are gone, your blood sugar is dropping precariously low.
So the brain is in serious trouble.
The brain is in trouble, and the body shifts into this emergency mode. The fat cells are dumping fatty acids into the blood. The liver is taking them up and burning them furiously through beta oxidation. The liver is absolutely flooded with acetyl-CoA.
A traffic jam of energy.
An energy traffic jam. And at the same time, because you're starving, the liver is desperately trying to make glucose for the brain via gluconeogenesis. And to do that, it pulls oxaloacetate out of the citric acid cycle.
Ah, so the citric acid cycle itself stalls because its key intermediate is being used elsewhere.
It grinds to a halt. So now you have a stalled cycle and a massive flood of acetyl-CoA inputs. The acetyl-CoA has nowhere to go. It just piles up inside the liver mitochondria.
A metabolic crisis.
It would be. But the liver has an escape valve. To relieve this pressure, it takes 2 acetyl-CoA molecules and sticks them together.
To form acetoacetate.
Exactly. And acetoacetate can then be reduced to beta-hydroxybutyrate. These 2 molecules, along with a small amount of acetone that's formed spontaneously, are what we call ketone bodies.
And the magic of these molecules is what?
They are water-soluble. They don't need albumin to be transported in the blood, and crucially, they have specific transporters that allow them to cross the blood-brain barrier.
So they are a soluble, transportable form of fat energy that the brain can actually use as fuel.
They are fat-derived sugar substitutes. During prolonged starvation, the brain can adapt its metabolism to get up to 75% of its total energy from ketones. And this is absolutely vital because it spares your muscles.
How does it spare muscle?
If we didn't have ketones, the only way to feed the brain would be to break down muscle protein into amino acids to make glucose. We would literally digest our own hearts and diaphragm to survive. Ketones allow us to preserve our essential protein and survive for months on our fat stores alone.
There's a really poetic detail in Harper's about the liver's role here. The liver makes the ketones, but it can't use them itself.
The altruistic organ. The liver expresses all the enzymes to make ketones, but it specifically lacks one key enzyme, often called SCOT, that's required to break them down and use them for energy.
It's like the chef who cooks this amazing feast but has no mouth to eat it.
It's an incredible fail-safe. It ensures that the liver doesn't selfishly consume the precious fuel. It just prepared for the brain and the other peripheral tissues. It exports 100% of it.
That is just incredible evolutionary design.
Yeah.
But systems fail. And when this particular system fails, it can fail catastrophically. Let's move to Section 6, the clinical aspects.
We've seen the beautiful efficiency. Now let's look at the pathology when a gear gets jammed.
Okay, let's start with a medical mystery. Jamaican vomiting sickness.
This is a classic toxicology case study. In Jamaica, there is a fruit called the ackee fruit. If you eat it when it's fully ripe and prepared correctly, it's a national dish. It's delicious. But if you eat it when it's unripe,
It's toxic.
it is dangerously toxic. It contains a compound called hypoglycin A, and hypoglycin is a potent metabolic poison. It specifically and irreversibly inhibits the enzyme medium-chain acyl-CoA dehydrogenase, or MCAD.
So it jams the beta oxidation grinder right in the middle.
It stops the process cold, specifically for medium-like fats. Now imagine the consequences. You eat this fruit, suddenly your cells cannot burn fat for energy.
Okay.
Your blood sugar crashes into a severe hypoglycemia because the liver can't get the energy from fat to run gluconeogenesis. You start vomiting, you have convulsions, and you can fall into a coma.
All because you've lost your backup generator. The fat-burning pathway is offline.
Exactly. It's a tragic illustration of how utterly dependent we are on this pathway, even when we don't realize it.
What about something like Zellweger syndrome? That involves peroxisomes. We haven't talked much about those.
Right. Most fat burning, as we've discussed, happens in the mitochondria. But some fatty acids are just too weird or too big for the mitochondria to handle.
Like?
Specifically, very long-chain fatty acids, VLCFAs, think 26 carbons or more, or branched-chain lipids like phytanic acid from dairy. The mitochondrial machinery says this is too big for me to process.
So where do they go?
They go to the peroxisome first. The peroxisome acts as a pre-processing plant. It has its own version of beta oxidation that shaves these massive fatty acids down to a manageable size, which can then be sent to the mitochondria.
So in Zellweger syndrome.
In Zellweger syndrome, children are born without functional peroxisomes. As a result, they accumulate these massive, weird fatty acids in their tissues, particularly in the brain and nervous system. It leads to profound neurological defects, liver failure, and early death. It highlights that every component of this system, even the niche ones, is absolutely essential.
Finally, we have to talk about the most common and probably the most dangerous presentation of this system going wrong. Diabetic ketoacidosis, or DKA.
This is the runaway train scenario. In a person with type 1 diabetes, the pancreas produces zero insulin. Remember our regulation. Insulin is the primary stop signal for fat breakdown.
So without insulin, the body thinks it's starving, even if the person just ate and their blood sugar is sky high.
The cells are starving in a sea of plenty. The fat cells get no insulin signal, so they scream starvation and start massive, uncontrolled lipolysis, dumping huge amounts of fatty acids into the blood.
And the liver gets this flood.
A liver receives this flood. With no insulin, malonyl-CoA drops to zero. The CPTI gate is wide open. The liver's beta oxidation pathway runs at a frantic maximal rate, producing an overwhelming amount of acetyl-CoA.
More than the stalled Krebs cycle can handle.
Far more. So the liver's only escape valve is to churn out ketones at a rate that far, far exceeds what the body can possibly use for fuel.
And ketones are acids, acetoacetic acid, beta-hydroxybutyric acid.
So the pH of the blood begins to drop. The body's bicarbonate buffering systems are quickly overwhelmed. The patient goes into a severe metabolic acidosis.
And you get the classic symptoms.
Kussmaul breathing as the body tries to blow off CO2 to compensate. Dehydration. A fruity smell on the breath from acetone being exhaled. It is a life-threatening medical emergency.
It really emphasizes the absolute necessity of that malonyl-CoA switch. DKA is what happens when that single regulatory switch breaks.
It's a system designed for resilience and endurance, but it requires incredibly tight regulation. When that regulation snaps, its own incredible efficiency becomes the enemy.
This has been quite a journey. I mean, we started with a sealed vault and a fat cell. We mobilized a key with a hormone. We took a minivan taxi, bribed a bouncer at the mitochondrial door, sliced a salami, and even created water for a hibernating bear.
When you step back and look at it all together, the theme that really emerges is integration. We study these chapters separately, glycolysis here, beta-oxidation there, but in the body, they are constantly in a dynamic dance.
The Randall cycle.
The Randall cycle, exactly. The concept that glucose and fat metabolism are in constant competition for access. When one is high, it actively suppresses the other. It is a beautifully balanced economy.
And it really puts our modern lives into perspective, doesn't it?
Yeah.
We carry this incredible machinery, this ultramarathon capability that allowed our ancestors to survive famine, to walk across continents. We are built to run on fat for weeks.
And yet, in our modern environment of constant food availability, of 3 meals a day plus snacks, we rarely let this system truly turn on. We live in a state of almost constant glycolysis. We rarely need to dip into the vault.
Maybe that's the final thought then. The metabolic fires are there, primed and ready for the long haul. The question is whether we ever give them the chance to burn.
Metabolism built for distance. I think that says it all.
Thanks for listening to this deep dive. We hope you look at that lipid droplet a little differently next time you think about energy.
Until next time, keep exploring.